Okay, so it's 9 o'clock. Let's get started. Welcome everyone to our today's museum seminar. And it's really my pleasure to welcome one of my old friends, also my colleague, Dr. Kairui from School of Life Science from Xiamen University, present his recent work today on our platform. So, as I just mentioned, Kairui currently is an assistant professor in School of Life Science at Xiamen University. Kairui complete his undergraduate study in Shandong Normal University, and afterwards he decided to pursue his PhD training in Institute for Biochemistry and Cell Biology in Chinese Academy of Science in Shanghai. And then after he completed his PhD training, he decided to stay in the same institute for additional 2 years, work as a research associate before he moved to United States, did his postdoc training. So from 2013, Kairui moved to NIH, actually with Ron Jaman in NIID, started his postdoc training until 2019. So during his postdoc work, he actually did a lot of groundbreaking discovery and then published a few high-profile paper. From 2019, Kairui started his independent research program, moved back to China, and then started his lab in Xiamen University. So Kairui's research actually mainly studied the regulatory effect of intestinal immune responses on host physiology activity. As the largest mucosal organ in the body, the intestinal tissue has a continuous interaction with the external environment. And at the same time, the intestinal tissue is rich in different immune cells, which play extremely important role in exerting barrier function and maintaining host physiology homeostasis. So his lab mainly use advanced imaging system and the traditional immunological method to study the activation of the immune system by environment factors, which including intestinal flora during physiological homeostasis and disease occurrence. At the same time, also, he studied the effect of different immune responses on the functions, physiological functions, such as gut microbiome development, epithelial stem cell differentiation, host metabolic regulation, and tumorigenesis in order to find new targets for establishing and maintaining the intestinal homeostasis and potentially develop new treatment approach for the gut disease. So I think today, Kairui probably gonna present us one of his newest discovery and work. And I think recently get just published. Congrats. And then we are very much looking forward to your talk, Kairui. Please go ahead.
Thank you. Thank you so much for your kind introduction, and also thank you for the opportunity to share our work with the fantastic audience through the Immune Zoom. Thank you. Today I will going to talk about the immune microenvironment from tissue to tumor. Okay, I will minimize this. Okay. Okay, as we know, for the multicellular organism, that the cellular organization is quite important, especially for the tissue functions. Different kind of type of cells can organize into the functional units of our tissues, such as the alveoli in the lung, nephron in the kidney, lobus in the liver, and villi in the small intestine. These functional units can exert the function of the tissue. And this is also true for the immune system, such as for the lymph nodes. There are B cell follicles and T cell zones in the lymph node, and also there's efferent and afferent lymphoid vessels. These vessels can be used as the transportation for the antigen into the lymph nodes, and also for the egress of T cells after activation. As mentioned by Chuan, I'm quite interested in the mucosal immunology and also for the gut. In our gut is colonized with trillions of the microorganisms, and they have lots of physiology functions to our physiology, and they help us digest the food and provide the nutrients. And also they can help us to defend against pathogens, education our host immune systems, and also regulation the nervous system. And recent literature also shown that the microbiome has also play also important roles in the tumor genesis and immunotherapies. Even though this microbiome has a lot of physiology functions, the dysbiosis can also cause a lot of disease, such as inflammatory bowel disease, the autoimmunity, and also are some related with nervous disease, such as AD and autism. To maintain the stability of the gut microbiome, the gut also accumulated with different kind of immune cells, include the innate cells such as the myeloid cells, the DCs, macrophage, the granulocytes, neutrophil, eosinophil, and also the innate lymphoid cells such as IRCs. And also there are adaptive immune cells such as the B cells and the CD4, CD8 T cells. And these cells are not evenly or randomly distributed in the gut. Most of cells can be organized into the secondary lymphoid tissue, such as the mesenteric lymph nodes and the Peyer's patch. And some of these cells are organized into the tertiary lymphoid tissues, such as crypt patch and isolated lymphoid follicles. I think these secondary and tertiary lymphoid tissue are very critical for their functions. As mentioned by Chen, I did my post-training in Ron's lab. We mainly use the multiplex advanced imaging tools to study the immune microenvironments in the gut. Here is the tertiary lymphoid tissues, as I mentioned, and there are different kind of cells accumulate together. This includes the rho gamma T positive, CD3 negative IRC3s, and also the CD3 positive T cells and the Tregs, and also dendritic cells and epithelium. There's lots of advantages to use the imaging tool to study the immune microenvironment. We can examine the cell states and the locations of different kind of cell types. From here, we can see that under steady states, there are very few KRG1 positive ILC2s in the small intestines. However, after treatment with IL-25, the ILC2 cells proliferate and they are become Ki67 positive cells. And also, some of the proliferate ILC2s can enter the circulation through the lymphatic vessels in the villi. And also, we can also include the functional molecular stainings in the imaging tools, such as to stain the phosphorylation of molecules. So we use the phosphorylation of STAT3 to imaging the gut. Under steady state in the wild-type mice, there's very few p-STAT3 signals in the small intestine. However, even also on the steady state in the rat knockout mice, there's tons of p-STAT3 signals. These signals are from IRC3s, the rho gamma T positive IRC3s, and all the epithelium. This means that the IRC3s and epithelial cells are constitutively activated in the absence of the adaptive immunity. And this kind of constitutive activation of IRC3 and epithelium start from weaning. So before weaning, there's no such p-STAT3 activation in the rat knockout mice. And in the wild-type mice, we also found that shortly before weaning at 4 weeks, there's transient activation of IRC3 and epithelium. And after the maturation of the gut adaptive immune system, this activation is shut down. Since this is already published, so I will make long story short. In the absence of adaptive immunity, some kind of the microbiota, such as the SLB, can activate monocytes to secrete IL-23, and IL-23 will activate ILC3 to produce IL-22. Then IL-22 will activate STAT3 in the epithelium to secrete the antimicrobial peptides to maintain the stability of the microbiome. With the maturation of the adaptive immunity in the gut, especially the T cells will shut down this kind of activation through different kind of mechanism. That Tregs can suppress production of IL-23 from the monocytes, and the Th17s can control the abundance of SLB through the neutrophil. Given that we have the innate and adaptive lymphocyte to control the microbiome, why we need such two complicated system? So we did the RNA-seq with the ILC3 and epithelium activation or not. We found that in the ILC3 activated mice, the metabolic associated genes are downregulated in these mice, which include the CD36, NPC1L1, FAB1, and FAB2. These are the lipid transporters. As we know, the main function of small intestine is to help the host to absorb lipids. So with the ILC3 activation, these lipid transporters are downregulated. As a result, the serum lipids are also reduced in the rat knockout mice. And also, as a consequence, the fat storage are also impaired in these mice. So from here, we can know that under homeostasis, the innate immunity and the function of gut, the lipid absorption are balanced. In the absence of adaptive immunity due to the constitutive activation of the innate immunity, the lipid absorption functions of the gut is compromised. Given the advantages of the imaging tools, after I set up my own lab, I decided to use these tools to study the tumor immune microenvironment, as the tumor immune microenvironment are very important for the tumor progression and also the efficacy for the tumor immune therapies. There are lots of literature about the tumor immune microenvironment. According to the infiltration of CD8 T cells, the tumor immune microenvironment can be divided into the infiltrated excluded tumors and also infiltrated inflamed tumors, such as the cold and the hot tumors. And also in some tumors, there's also tertiary lymphoid tissues constituted by the T cells and the B cells. Even we have already some advances in the tumor immune microenvironment, but there are still some basic questions, such as what's the positioning of the key immune cell populations in spontaneous mouse models? How the tumor immune microenvironment is organized? These basic questions are still unknown, especially for the detailed organization of different kinds of immune cells. So we decided to use colorectal cancer mouse models. This is the APC-mean models. This is a spontaneous tumor model. And also APC is a tumor suppressor genes. It can suppress the Wnt signaling that is critical for the homeostasis of the gut. And in the human colorectal patients, this is the APC gene is the most prevalently mutated genes. And there are 51% of patients of the hypermutated tumors have this APC mutation. And also 81% of the people for the non-hypermutated tumor have the APC mutations. So the APC gene is considered as the initiator of the colorectal cancers. So we use the APC model to study the immune microenvironment. At the beginning, we want to know where is the tumor. From the tissue, we can see the tumor, but under microscope, where is the tumor cells and where's their stromas. So I used the Ki67, the proliferation marker, to show where's the tumors, because the tumor cells are the constitutively proliferating cells. So in the wild-type mice, the Ki67 cells are at the crypts for the intestinal epithelium stem cells and also the transient amplifying cells. And in the tumors, most of the tumor cells are proliferation. So by using the Ki67, we can distinguish the tumor region and also the tumor stroma region. Next, we studied different kind of immune cell populations, such as the macrophage, DC, B cell, CD8, CD4, and Tregs. From here, we can see that there's very few CD8 T cells in the APC mean tumors, and also very few B cells. And for the Tregs, we can see that there's very high abundance of Treg cells in the tumor stroma region. And for the macrophage and dendritic cells, they are evenly distributed in both tumor and tumor stroma. If we zoom in, we can clearly see that majority of the Tregs are accumulated at the tumor stroma, but not in the tumor. And interestingly, we also found that majority of the Treg cells are positioned along the vasculature. So we studied the line 1 for the lymphatic vessels. We do see that majority of the Tregs are positioned along the lymphatic vessels. This is a small tumor. When the tumor progression, we can see that the Tregs accumulate at the lymphatic region more obviously. And also we did the 3D imaging to show how the Tregs positioned along the lymphatic vessels in the tumor. Here you can see there's lots of the lymphatic vessels, and majority of the Tregs sit accumulated at the perilymphatic regions. And also, besides the lymphatic vessels, blood vessels are also vasculators in the tumor microenvironment. So we compared the location of Tregs to both lymphatic vessel and blood vessel. From here, by quantification, we can see that the Tregs are more closely associated with the lymphatic vessel compared to the blood vessels. So from this data, we can see that regular T cells positioned as the perilymphatic region in the tumor stroma in the mouse colorectal cancers. After getting these findings, we can raise at least 2 questions. Why Tregs positioned as the lymphatics in the tumor stroma? What are the physiological functions of Tregs with this special location? These are 2 questions that I think everyone is quite interesting. But these questions are not easy to answer at the beginning. So we started with another question. What's the phenotype of the tumor Tregs and their microenvironment in the tumors? So we did the bulk-seq. We isolated the tumor Tregs and the colon Tregs and did the bulk-seq. We found that loss of T cell activation-associated genes are upregulated in the tumor Treg, such as the PD-1, OX40, TIGIT, CD69, and also ICOS. So compared with the colon Tregs, the tumor Tregs has the activated signatures. We also confirmed this expression at protein level by flow cytometry. From here, you can see the tumor Tregs indeed express higher level of PD-1 and TIM3. But this higher expression is not homogeneous. And some of the tumor Tregs express higher level and some has the lower level expressions. So there's a heterogeneity of the Tregs even in the tumor microenvironment. So next, we also did the single cell RNA-seq of all the immune cells from the tumor and the tissue. We can see that every immune cell population are discovered in our single cell RNA-seq data, include the B cell, neutrophil, T cells, macrophage, DCs. If we zoom in the Treg cells in the tumor, we can see there's at least the two populations of the Tregs, the activated Tregs and also the quiescent Tregs, distinguished by the signature genes of T cell activation, such as CTLA4, ICOS, PD-1, KRG1, TIM3, LAG3. Since the tumor Tregs has the activated signatures, we want to know which cell population is responsible for the Treg activation. Even though Tregs as a subset of the CD4 T cells, their activation depend on the antigen presenting cells, such as DCs. So we analyzed the DC populations in the tumor. We found 4 clusters, such as the CDC1, CDC2, PDCs, and MRAG DCs. I think all the audience already know the CDC1, CDC2, and PDCs. And for the tumor immunologist, MRAG DCs are just recently discovered cell clusters. These cell clusters are characterized by higher expression of CCR7, CCR22, and PDR1. This population is not the first discovered in our study. In about 2019 and 2020, there are at least 3 groups discovered these DC clusters in different kind of tumors, such as the lung cancer and the liver cancers. This population, these DC clusters has very high maturation status. They express higher level of CD80, CD86, CD40, and also they express higher level of CCR7. So they have the migratory potential. And also they can secrete higher level of CCR17, CCR22. Besides these activation and maturation signatures, the MRAG DC also have the regulatory signatures. They express higher level of PDR1, PDR2, and also SOX genes. So that's why they are named as the MRAG DCs. With the single cell RNA-seq data, we did the ligand and receptor pair analysis to examine the potential correlation between the Tregs and DC clusters. From here, we can see that the activated Tregs are more associated with the MRAG DC, such as the chemokine and chemokine receptor, CCR4, CCR22, and also the T cell activation and DC maturation markers, such as CTLA4, CD80, PD-1, PDR1. So from here, we can know that regular T cells in the tumor exhibit activation signatures and are highly correlated with the MRAG DCs by transcriptome analysis in the mouse tumors. This is just through the transcriptional analysis. So what's the spatial relationship between Tregs and MRAG DCs? So we use the CCR22 TD tomato reporter mice to examine the MRAG DCs in the APC mean mouse tumors. From here, we can see all the CCR22 T tomato cells express higher level of CD11Cs. This means that these cells are really MRAC DCs. And we can see from this imaging that the MRAC DCs and Tregs are highly associated with each other as the perilymphatic region. So I think the Treg MRAC DC lymphatic niche is established in the tumor stroma. Next, we also want to compare whether the Tregs association with the MRAC DCs are specific or not. So we compared the association of Treg cells with the CDC1 and CDC2 by using different kind of markers. We use CD103 MHC class 2 positive to indicate CDC1 and CD11B and MHC class 2 F480 negative as the indicator of CDC2. From here, we can see that the MRAC DCs are more associated with the lymphatic vessel and Tregs. And also, the Tregs are in closer proximity to MRAC DCs than to CDC1 and CDC2s, which means the Tregs are more specifically associated with MRAC DC at the perilymphatic region. As I mentioned earlier, that the tumor Tregs has the activation heterogeneity. Some are activated, some are quiescent. Whether these differences are also associated with their microenvironments. So we also stain the PD1 together with FOXP3 and by using the CCR22 TD tomato epithelium mice. We can see that the PD1 and OX40 expression on the Tregs are highly associated with MRAC DCs, which means the closer of the Tregs to MRAC DC, the higher expression of these molecules, PD1 and OX40. So this means the activated Tregs are highly associated with the MRAC DCs. To further investigate whether these Tregs are activated locally or somewhere else, and then migrated to the MRAC DC in the tumor microenvironment. So we used the NER767 reporter mice. The NER767, as we know, is the early or proximal TCR signaling. With this kind of expression, we can know the TCR signaling in situ. So from here, we can see for the Tregs associated with the MRAC DCs, some of them are the NER767 GFP positive cells, which means the Tregs are locally activated on the MRAC DCs. Given the antigen presentation by the MHC class 2 molecules is very important for T cell activation, we also want to know whether this antigen recognition is important for the Treg activation. So we injected the anti-MHC class 2 antibody in situ into the tumor and examine the PD1 expression by the Tregs. From here, we can see with the control antibody treatments, there's loss of PD1 high Tregs. After anti-MHC class 2 antibody treatments, the PD1 expression dramatically reduced, even though these Tregs are still associated with MRAC DCs. So from here, we can see that a Treg MRAC DC lymphatic niche is established in mouse colorectal cancer, and this kind of niche will facilitate Treg local activation. So next, we want to know what's the molecular mechanism for the establishment of such niche. We started from the phenotype of the MRAC DC. As I mentioned earlier, that MRAC DC exhibit maturation state and they express higher level of CCR7. And CCR7 is a homing chemical receptor for their migration to the draining lymph nodes through chemical gradient. such as the CCL19 and CCL21, which secreted by the lymphatics. And also, the MRAC DCs can secrete chemokines such as CCL22 and CCL17. These chemokines can recruit Treg cells through CCR4. So we suppose if these hypotheses are correct, then it can explain how this niche is established. So we first stain the CCL19s in the tumor stromal. We do see that it's the lymphatic endothelial cells, but not the blood endothelial cells express CCL21. This can explain why the MRAC DCs could migrate to the lymphatic vessels because they express the CCL22 receptor CCR7. And also we want to know whether CCR4 is also critical for the niche formation. So we transferred the wild type or CCR4 knockout Treg with different fluorescent protein labeled Tregs into the APC mice. After transfer, we examined the Treg distribution in the both mesenteric lymph nodes and tumors. In the mesenteric lymph nodes, the both wild type and knockout CCR4 are kept at 1-to-1 ratio. However, in the tumors, there's very few CCR4 knockout Tregs, which means without CCR4, the Treg cell migration into the infiltration into the tumor are impaired. To further examine whether these Tregs can be recruited to the MRAC DCs, to the niche, so we use the mixed bone marrow chimerics by transferring the CCL22 TD tomato bone marrows together with CCR4 knockout ubiquitin GFP bone marrows. By using the CCL22 reporters, we can visualize the MRAC DCs. And also by using the CCR4 knockout ubiquitin GFP, we can see the CCR4 knockout Tregs. And the GFP negative Tregs should be derived from the CCL22 reporter bone marrows. So in the tumor microenvironment, we can see the MRAC DCs. And also we can see the Tregs are associated with the MRAC DCs at the peri-infected region. If we examine the origin of these Tregs, we can see that the majority of the Tregs are GFP negatives. And for the GFP positive cells, they are distant to the MRAC DCs. And also we examine the activation of such Tregs in the niche. From here, we can see that within such niche, majority of the Tregs express higher level of PD-1, the yellow markers. And also only one Tregs from the CCR4 knockout bone marrows, and they didn't express PD-1. Next, we also want to know whether this niche is also important for the tumor progression or tumor regression. So we specifically delete the CCR4 in the Tregs by using the FOXP3 CCR4 flox mice and cross these mice to the APC mean background. We see that in the control mice, there's lots of tumors in the small intestine. However, in the absence of CCR4 on the Tregs, there's very few tumor and the size of tumor are very small. So from here, we can know that the CCR4 is essential for the establishment of the Treg and MRAC DC lymphatic niche, thereby promoting Treg activation and suppress the anti-tumor immunity. So what's the mechanism for the Treg restraining anti-tumor immune response? As we know, there's several steps for the successful anti-tumor immunity. The first step is the uptake tumor antigens by the dendritic cells. Upon uptake tumor antigens, these dendritic cells become mature and they express CCR7 to migrate to the tumor-draining lymph nodes and present the tumor antigen to the T cells. After T cells recognize this kind of tumor antigens, these cells will activate and proliferate. These activated and proliferated T cells will migrate into tumor region and execute their effector functions. So for a successful anti-tumor immunity, the tumor antigen presenting is a very critical step to initiate the anti-tumor immune response. Given that the MRAG disease has all the characters that's responsible for the tumor antigen transportation from tumor to draining lymph nodes, so we examined the DC populations in the mesenteric lymph nodes of wild-type mice and APC mean mice. We can see here that with the tumor, the migratory disease in the draining lymph nodes are decreased. And there's reduced MRAG DC, but increased number of resident disease in the tumors, in the mesenteric lymph nodes. Given that only the tumor antigen bearing migratory disease is responsible for the anti-tumor immunity, so we are more interested in the tumor antigen bearing migratory disease in the draining lymph nodes. Given we use the genetics models, so it's very hard to get to visualize the tumor antigens. So we injected the fluorescent labeled ova into the tumor or into the tissue to mimic the tumor antigens. And 24 hours after in situ injection, we examined the PD-positive MRAG disease and resident disease in draining lymph nodes. From here, we can see if we injected PD ova into tumor of APC mean mice, there's very few PD-positive migratory disease in the draining lymph nodes. If we inject such antigens into the tissue, there's lots of PD-positive MRAG disease, which means the transportation of tumor antigens to the draining lymph nodes are impaired in these mice. As contrast, the PD-positive resident disease are comparable between these mice. This means the PD ova are transported by the antigen presenting cells, but not the diffuse of these antigens. In order to further analyze the anti-tumor immune response, we also transferred the OT1 or OT2 cells into the wild type and the APC mean mice, and in situ injected ova plus LPS. And we also blocked these T cells in the draining lymph nodes by using the S1P inhibitor, the FTY720. After 5 days, we analyzed the T cell proliferation. All the OT1 and OT2 cells are labeled with the fluorescent dyes. From here, we can see that in the APC mean mice, the proliferation of the OT1 and OT2 cells are dramatically decreased. And also there's decreased number of these T cells. So which means the T cells priming in the tumor draining lymph nodes are impaired in the tumor-bearing mice. In order to know whether this kind of impaired anti-tumor immune activation is due to the Treg and MRAG DC lymphatic niche. So we deplete Tregs by using the FOXP3 DTR mice and administered with the DTX. So after Treg depletion, there's dramatically increase of the tumor antigen transportation to the draining lymph nodes. And also because we know that depletion Tregs has a dramatic effect on the tumor microenvironment. So we want to specifically disrupt the Treg and MREG DC crosstalk by using an undepleting anti-CTLA-4 antibody. After blocking the crosstalk between MREG DC and the Tregs by using the anti-CTLA-4 antibodies, we can also see an increase of the tumor antigen transportation to the draining lymph nodes. So next, we want to know why there's increase of the tumor transportation to the draining lymph nodes. So we examine the MREG DC phenotypes after depletion of Treg or anti-CTLA-4 treatments. From here, we can see that without treatments, the MREG DC express higher level of CCR7, CD80, and CD86 compared to the CDC1 and CDC2 populations. After depletion of Tregs, the expression of these molecules are furtherly increased. So which means with after depletion of Tregs or blockade of the Treg and MREG interactions, the MREG DC has higher capacity to migrate to the draining lymph node and present antigens to T cells. So from here, we can know that the Tregs can suppress the initiation of anti-tumor immune response by restraining tumor antigen trafficking to tumor draining lymph nodes. So given all these studies are done in the mouse models, so does the Treg MREG DC lymphatic niche also exist in human colorectal cancers? So we stained the clinical samples with the imaging, and we found that majority of Tregs are also located at the perilymphatic region in the human samples. This is inconsistent with our findings in the mouse. And also, we analyzed the TCG database and found that the Treg and signature are highly correlated with the MREG signatures in majority of the cancer types, include the colorectal cancer, and also the breast cancer, the lung cancer, and also sarcomas. So we want to know whether the Tregs and MREG DCs are also associated with each other, especially in the human samples. So we analyzed the published spatial transcriptomic data in the colorectal cancer. From here, we visualized the activate Treg signature and MREG DC signatures, and also the lymphatic signatures. From here, we can see that the activate Tregs are co-localized with the MREG DCs at the perilymphatic region in the human samples. And also, the activate Tregs are positioned in closer proximity to the MREG DCs compared with the quiescent Tregs. So from here, we can see that the Treg MREG DC lymphatic niche are also exist in human colorectal cancers. So what's the physiological relevance of such organization of the immune microenvironment? So we divide the colorectal, we analyze the TCG database by divide the colorectal cancer patients into 3 groups. One group is the MREG DC signature low group. The other 2 is the MREG DC high group, and separated by the activate Treg signatures, the MREG DC high, activate Treg low, and the activate Treg high populations, and analyze their survivals. From here, we can see that with higher signature of MREG DC and low signature of Tregs, especially active Tregs, has better survivals in these populations. And this is true for the disease-free and progression-free survivals. So from here, we can know that the Treg and MREG-DC infect niche is also established in the human colorectal cancer and can be used to predict the survival outcome. So from this study, we found a specific suppressive niche in the tumor stroma. That's the Treg MREG-DC lymphatic niche, which is critical for the anti-tumor immunity. Within this niche, the Tregs acquired activated signature, and then they restrain the tumor antigen transportation to the draining lymph nodes. So this is our findings. So at last, I will thank my students, Yu Siyuan and Li Shuqing, for this great work. And also, I will thank my collaborators, such as Han Jiahuai, Deng Wenbo, Yu Rongshan, Ren Jianling, for their help. And also, I would like to thank Dr. Qi Hai from Tsinghua University for the help. And also, I will thank the grants. So at last, I will make an advertisement through this platform. All the postdocs and the PhD students' positions are available at the lab now, and welcome to join the immunophysiology lab at Xiamen University. Thank you. Thanks so much.